Okay, thank you, Rick. And good morning, everyone. It's our last day today for the symposium. Uh, Today, I want to welcome our speaker, is Dr. Ben. When I saw his profile, it's uh, very distinguished profile because it must make our LinkedIn written in the crayon. So Dr. Ben, he is head and vice president of clinical and quantitative pharmacology in Adagene, and you are which is a biotechnology. It's for focusing on cancer. Dr. Ben, he take his biography and he start his journey in University of Sichuan, which is take a biological science. And then he moved to University of Washington and take his PhD in pharmaceutical science. Because why just stop in the one degree? You can collect them like a Pokemon cards. But and also he become one of the gator. He joined University of Florida as a postdoc, and also he become intern in Seattle Genetics and serve as honoris fellow at FDA. So if you are feeling accomplished, you can join my club. So Dr. Ben also he authored about around 30 reviewed publication and book chapters, and about he co-invented about 10 patents. I am not saying he is a superhero, but I have never seen him with Superman in the same room. So with Batman in the same room. So yeah. So also he served as editorial board of clinical pharmacology and therapeutics and translation science, and also he is affiliated as associate professor at the University of Washington and adjunct assistant professor at the University of Florida. Basically, he is shaping of mind of the next generation of scientists. So please welcome Dr. Ben and hope you join his presentation.
Thank you for your nice introduction. All right, so I think today, right, again, I want to thank Sarah for inviting me. And when I was thinking about what type of presentation I'm going to give, I'm going to give a very unconventional one, as you see. I'm going to set my timer here 30 minutes because I would really want to be on track. So, okay, so this is actually basically the recap of the intro. I think I want to talk a little bit about this because you will see how some of the experiences really shaped up the decision-making process down the road. For example, right, like one thing I did not put into the introduction is really back at the University of Washington as an exchange student, I worked on erythropoietin, which is actually an approved biologic, but I never knew that I was going into the Janssen Biotherapeutics, which actually made one of the recombinant human erythropoietin back in the days. And what I did at University of Washington for classic enzyme kinetics, how that leads to some of the work I'm doing at Adagene, which also make antibody-based therapeutics using mass technology and also enzymatic cleavage. So I think there are a lot of interesting connections along the journey, which I will shed light upon for some of the case studies, which you will see. So let's move forward. For this audience, just a very quick introduction. So back in 2020, working with Brian and Yen at Janssen Biotherapeutics, and we actually contributed to a paper to the pharmacy textbook, Remington, for the first time introducing some of the biologics-related components. I would have to say, I think, as you just heard, right, from Rich, the new modalities, bispecific ADCs, FC fusion molecules, and also FC engineered molecules have really, like multi-specifics have really revolutionized, I think, the antibody-based treatment. Of course, biologics, right, really capture a wide range of products, which I think for today, I'm going to touch upon a few of them. So one thing I was thinking about to kind of go into the case study is really some of the questions I've been thinking the last couple of years for what the pharmaceutical and biotech industries are faced with, right? So to name a few, I think at least in my experiences, part of the key challenges, how do you develop a next generation or new molecule, best-in-class type of molecule that can really differentiate from existing molecule? How can you predict a POCs, right, a success early, particularly even in the preclinical setting or in the phase 1 setting? Of course, a lot of the phase 3 studies, right, still suffer from failure, even though you may have a phase 2 success. That's a different story, which I'm not going to cover today. One other thing is that, again, combination therapy, bispecific, multispecific, right? How do you justify the approach for bispecific instead of combination approach? The other thing is about dose selection, right? Dose selection, Project Optimus, you've heard about it. Really for molecules either with existing marketed compounds or with a first-in-class type of mechanism. I think it's really important, given that we really want to make sure that patients, right, get the best drug at the appropriate dose that maximize their therapeutic benefit without significant safety concerns. So here is one interesting diagram that we came up with in this particular Remington book chapter. It does capture some of the key consideration, at least from my side, looking into how biology, pharmacology, biophysics, together with non-clinical PK/PD, together with non-clinical safety study, and also the translational sciences, particularly leveraging existing information from the public domain, from other companies, for example, and to build that human dose prediction, and together with the clinical finding through the clinical study, how does that really provide reverse translation opportunity to enable better second generation or next generation molecule making? I'm not going to go into the specifics for some of the consideration here, but as you will see, modeling plays a very central role to actually guide not only the initiation of a project in terms of the therapeutic concept, but also how you can guide the project team towards designing the best and fit for purpose studies, right? Animal study, even in vitro study, to give you the data set and also work with the analytical scientists to generate the proper assay to allow for the program to move forward with the best efficiency. So I think really, again, modeling and simulation is a very critical part along the whole development path from very beginning of a product initiation. We talk about concept, right, called the target product profile all the time. But I think the other thing is really the target construct profile, right? What are the key properties you want to hit? I think that's something we don't have a lot of opportunity to talk about, but I want to touch upon it. So I have a 3 set of case studies. The first case set is really related to the physiologically based pharmacokinetic modeling, because that's where everything started, at least in terms of modeling journey. So the first case is really this particular one, which I worked on in Seattle Genetics back in 2010, the summer of 2010, as my internship project, to develop a novel PBPK model for an anti-CD70 ADC in tumor-bearing mice. So Baiteng was my supervisor back then. At that time, there's no published ADC PBPK model, full PBPK model. But fortunately, there was this very nice paper from Dr. Balthasar looking into FCR and knockout mice, right? So what we did is that we leveraged the information from that 2006 paper, if I recall correctly, and built in the component of the toxin together with the tissue level PBPK, particularly within the tumor, which we saw somewhat different kinetics compared to some of the other organs, right? For example, we did collect a wide range of organs in those tumor-bearing mice. And we do have a measurement in terms of the concentration, for example, ADC, and also the toxin, the free toxin in serum, as well as the values, I believe, in the tissue. So I think the challenge we had is that SynBiology MATLAB was not a thing back then, and I had to actually write the over 60 differential equation in non-man, which was not running in the beginning, but thankfully, Baiteng was able to spend 30 minutes with me every day within that 2 months or so for us to actually come up with this model that successfully converged, and then we presented that at the ACOP in 2011. So I think really that set the foundation for me to really think about, okay, what can we do in terms of the modeling approach? Even though I don't know how to use non-man, but really with a proper mentor, the question seems to almost impossible to tackle back then became fairly straightforward looking back, right, what we did back then. So moving forward very quickly for another 10 years, I want to talk about this particular study. This is to use a physiologically relevant mice colitis model to really understand the induction dose in IBD. Again, right, I remember one of the question come from day one is that why do we use induction dose? And as you guys know that J&J have a few approved molecules, right, in this space, and we were interested in understand the mechanism behind it. So what we did, right, before the whole modeling work is that we look into literature to see what are the relevant animal model that can really mimic human disease, right, in terms of physiological changes and also the therapeutic effect in terms of the drug that can be used. So we landed on using this particular CD45 RB high adoptive T cell transfer model. As you can see, this is a massive mice study with a lot of mice, and we actually use PK modeling to guide this very sophisticated dose fractionation study, looking at different schedule, different dose in mice. The most important finding from this particular paper is that you can see that the serum-free TNF-alpha concentration actually had a pretty weak correlation in terms of the full colon histopathology score that we some different readouts. But if you look at the colon-free TNF-alpha, again, each sample represents one mice, right? You do see that the correlation is much better. So really, this suggested that when you look into for example, some of the anti-ligand antibodies, you need to zoom into the site of action. But in order to fully explain this study, the key question is that how can we develop a mathematical model to really understand not only the antibody tissue distribution, but also the target suppression, target engagement at the site of action? So what we did, I want to highlight Neojin here, right? She was my intern back in when we were doing this project. We used the modelings to come up with this fairly sophisticated model that leveraged the minimum PBPK model was also initially developed, right, at Buffalo by Dr. Cao and Dr. Jasko back in like 2012 or so. And as you can see that this model was able to capture the measured serum PK, colon PK for the antibody, as well as the serum-free TNF-alpha and also colon-free TNF-alpha across all the different dose groups that we designed. Without going into the detail, what I would say that this study really pinpointed the importance of understanding target site, target suppression by an antibody. And again, right, we have a little bit more description towards why the loading dose was important. It's really able to suppress that initial flare of the disease within the colon and really have the TNF-alpha level down very quickly. So actually, if you look at the approved IBD-related molecules, right, regardless of the mechanism of action, they generally have a loading dose approach. I think the concept's fairly similar. But this really was, again, right, one good example to really show that without proper study design, proper analytical assay being developed, you would not understand that. One last thing is that when we talk about cytokine, right, there's a pretty good review paper looking to different cytokine and talking about cytokine circulation from tissue to circulation, as well as local consumption by cells. TNF-alpha is one of those cytokines that actually don't get represented at the circulating level because they do have a lot of local consumption by cells. It's a very important immune mediator. I would encourage you guys to look into this if you're interested, or talk to me later. So the third example, I want to really highlight the work, right? Viva and Dawei and Tom led for this very nice siRNA novel model that we worked on. I think again, without going to some of the project-related specifics, right? Really, we want to understand how does a PBPK model, right, develop for, like, for example, like a map can extend to a little bit more novel therapeutic modality like siRNA. So we actually use literature data looking into this particular anti-thrombin siRNA. The cool thing that Viva did, right, leveraging his extensive training in his PhD, he was able to build in some of the detailed MOA part for siRNA and leverage the data in mice, looking into, for example, total liver PK, the risk-loaded siRNA in the liver, and also the changes in terms of the mRNA for the liver AT, which is antithrombin, and also the serum AT level, and then predict how this would look like in human. Again, we use clinical data, which was shown here, digitized, to really show that you do have translational utilities from mice to human. Again, this paper was later used to understand other approved siRNA. So I want to just use the story to really show that, again, right? I think when you think about the PBPK framework, it does not matter what modality is. The important thing is that you can really capture some of the important physiology that a traditional complement model may not capture, that allows you to do cross-species comparison and also have a little bit more predictive power when you want to go into untested dosing scenarios. So the last study for the PBPK is really some of the work I'm currently leading within my current company. As I said, right, Adagene, we have the same body technology, which utilize a protease cleavable linker together with masking peptide. It's able to be selectively cleaved, for example, in tumor microenvironment where the enzyme level have been increased across a lot of different cancer types. So we presented the work, right, in SITC in last year and also ASCO. Really, you can see the simple PBPK, minimum PBPK diagram here. What's not shown here is really how we are able to incorporate the more sophisticated enzymatic cleavage, right, within the different parts of the model, and also account for what we know about SafeBody technology into the mice model fitting together with the human data from our phase 1 study, and really correlate with individual level patient response. Even though I think this model, right, is going through a few more generation of iterations, but I do have to say, without the prior experiences, right, with the minimum PBPK, the full PBPK, and also some of the enzyme kinetic learning at UW, I don't think I can really do this with this very sophisticated approach. So case that 60 minutes away. Case set 2 is I'm going to talk a little bit more about a different sets of journey with the TMDD model in a non-PBPK framework. I think PBPK has its own advantages, but the problem is a lot of times you don't really have the data to really inform the model building. So this is still considered to be, I would say, the gold standard for a lot of the questions that can be readily addressed. So I want to start the work that Stefan and I did back at my postdoc at University of Florida. So the question is that for MAPS with TMDD, what is the appropriate dosing strategy in children? I think the question, again, right, this is a project that Amgen was very interested in. And what we did first is we look into all the approved antibodies with indications approved in children. Out of the 9 molecules that actually have TMDD, right, a nonlinear PK, we found the majority of them have either body weight-based dosing or body surface area-based dosing. And even the omalizumab, which is the anti-IgE molecule, has both the baseline for total IL-6, IgE, sorry, and also the body weight at a different tier. So I want to show one example that we actually listed here, looking into the same target concentration utilizing body weight-based dosing. So Puneet Puneet is the second author of the paper, who was working with us back then as well. We had this fairly systematic approach, looking to same target concentration between adults and children, and the same target amount in terms of the adult and children. And we evaluated both dosing approaches and also full TMDD model together with the mechanism approximation of the TMDD model. So what I want to say in this particular case, right, looking back at more than 10 years now, even though we did not have one molecule that has gone through the actual measurement in terms of the target concentration characterization between adults and children, but the example, right, that we used for this particular theoretical exercise was based upon the anti-ILK antibody, which again, they have a very nice publication back in 2011 or so using actual human data. So we used that particular membrane-bound targets PK profile in human, and then really come up with an approach to really say that if the adults and children have the same target concentration, when you, for example, think about the TMDD framework, right, within the tissue, then really body weight-based dosing is able to give you a fairly similar proportion in terms of the the TMDD-related clearance versus the total clearance, and also importantly, over days, a fairly similar target occupancy over time. It's not exactly the same, but I think at least that this provides a framework. And I would have to say that I think this is still an area of further research, right? Understanding for specific target, is there any change over time? I don't think it's fully understood at the experimental level yet, but I think this is one area that needs a little bit more work. At least the groundwork was done here. So moving forward, after joining J&J, one of the first project I took on, right, is to work with Wei Rong, my manager back then, to evaluate for a fast turnover soluble ligand, how does things work, right? So I think what's really interesting about this particular project is that we started with the IL-6 model, sorry, IgE model. IgE is a much slower turnover target compared to IL-6. So we used our internal data for this anti-IL-6 antibody called the siltoximab. And what we did is that we actually, again, right, measure 3 different analytes: total drug, total target, and free target. And you can see that when we change the in vivo KD value to be at different values, we actually were not able to capture the free drug concentration for free IL-6 after the dosing, even though we can capture, for example, the total drug and total target very well. So through likelihood profiling, to understand the parameter identifiability, right, using the same model structure that we use for IgE, we found that, interestingly, the total drug and total target data only for a fast turnover target may not give you the same level of, I would say, data for you to really dissect the in vivo KD value and the elimination rate constant of the free target. I think that the work then was still pretty preliminary. I think there was a lot more work done in terms of understanding, right, the parameter identifiability for TMDD model. But at least this is an initial attempt to really show that understanding the free target measurement post-dosing for a fast turnover target is actually quite important. I mean, one of the biggest issues, right, a lot of the fast turnover target also have very low baseline level. And it really requires a very sensitive assay. But I think this work, right, prompts another important piece of work, which I also did back in the 2014 timeframe, and contribute to my first patent, right? Co-inventorship is really to leverage all existing data, particularly Amgen published the paper for etanercept, right? Another anti-TNF alpha, not an antibody, but a smaller construct, to really show that it has utility in terms of treating children with a new onset type 1 diabetes. So, but back then, Symphony was not used at all in children for this particular indication. So what we did is that leveraging the population PK model, which we have a lot of data for, and together with what we can collect in terms of the binding affinity differences, the PK differences between some golimumab, right, that's the J&J's molecule, and then we were able to come up with a very interesting simulation for induction period as well as maintenance period dosing, and we use this particular modeling approach, right? And then this is the final studied dosing regimen in children. And the paper was eventually published in 2020, right, in New England Journal of Medicine, and really showed that golimumab was able to result in better endogenous insulin production and less exogenous insulin use than placebo in this particular patient population. I think, again, right, this was really a very good example of how you can leverage both internal data as well as external data to do something that's really beneficial for patients. So, moving forward, I want to kind of like 10 minutes. I want to talk about the study 8, which is there's a very interesting bispecific project. Again, kind of go beyond just anti-TNF alpha, but we have this bispecific monovalent arm binding for TNF alpha as well as interleukin 17A. So for this particular publication, right, what we focus on really is to understand, does in vitro KD value predict in vivo KD value for this particular construct? So what we found most strikingly is that we've heard enough time, right, that for antibody that targeting soluble targets, you should be able to use your in vitro KD to, for in vivo. But the particular learning from the study is that because this monovalent binding, even though in vitro it does not really show differences in terms of, for example, using SPR, right, compared to their bivalent parental molecule. But when we look into our measured data in cyno monkey, utilizing some of the two molecules that we have, right, parental molecule and also the bispecific, that we captured the TMDD profile for both TNF alpha as well as IL-17. One thing I'll have to notice that for IL-17, we also were able to measure the free IL-17 here, but for TNF alpha, the level was so low we could not measure it. But we were able to measure these two. But I think, again, right, I want to talk about the caveat for the TNF alpha part of the study. So you can see that without any model estimation for human data, we directly leverage allometric scaling to predict what may happen in human and overlay with our human data that came from the healthy subset study in phase 1. So for IL-17, you can see that the prediction was almost spot on across the different dose level. But for TNF alpha, actually the prediction was not so great. But nevertheless, what we found is, I think the most important thing here, right, is that if you look at another molecule, Humira, adalimumab, at 1 mpk, right, which was equivalent to this particular dose level that was studied, 40 mpk, this is a 1 mpk for the bispecific, but 40 mg flat dosing sub-Q for Humira. It's able to result in a much higher total TNF alpha concentration, representing better target engagement compared to this particular monovalent bispecific. So the key thing is that we knew that this may happen in human once we get our model fitting down in cyno monkey. The project team decided to move forward the project to go into human because we really want to see, right, what may happen there. And really from this particular set of assessment, we felt that this molecule is not able to engage those targets as effectively compared to, for example, like a combination approach when you use bivalent antibody. And not to mention, right, you can not really compete, for example, even like approved molecule. So I want to quickly move to the set 3, which is really the PD modeling and also other types of mechanism-based modeling. I'm going to go back to my Adagene next time. The second project I did with Byton, right, is really this particular one that where I got to meet Rob for the first time and also Dr. Don Major back in AAPS NBC meeting back in 2011 in San Francisco. So what we did back then is we also want to understand how can we predict human doses, right, for ADCs. And fortunately, the Simone CDM and also SDM model was already published back then in 2004. So the challenge we face is that we were seeing nonlinear PK and also dose-dependent absorption profile for the particular ADC, but we were able to incorporate that into the PK part of the model, and we were able to use SDM as well as the CDM model for 2 different xenograft mouse model, and then understand what is the tumor static concentration, which was further used, right, for guiding human dose selection. So that was actually a very, very important learning for me besides the PBPK model. And then fast forward in 2015, we presented, right, at ASCR, this particular approach for evaluating a number of different antibodies, for example, cetuximab, trastuzumab, panitumumab, and also onartuzumab, EGFR, HER2, and c-MET, right? So I digitized data from a number of publications. Again, this particular framework was already published in 2013 by Nahor, right, back in the days, and leveraging mouse PK, xenograft efficacy, mouse PK/PD together with human PK prediction by cynomonkey, you can really do that for human. So what we did is that we actually did actually model feeding to the mouse data we digitized. We were able to predict the human dose, right, for those molecule, including bevacizumab, which is anti-VEGF, within 2-fold of their approved doses, or at least for this particular molecule, their later stage trial cases. And this particular methodology was further utilized, right, to support the first human dose selection as well as the RP2D selection for a recently approved molecule, which I'm still working on the publication with Wei Rong. So very quickly, switching gear, I want to touch upon this particular piece of study. The key question is, can we estimate a molecule-specific property, which we coined cross-arm binding efficiency for a bispecific antibody, looking into how the second arm binding is facilitated by the first arm binding that cannot be readily measured experimentally, and also assess its functional importance, right? Again, amivantamab is the drug that was approved with accelerated approval back in May 2021. And we have a number of 2 molecules that allow us, right, to really assess the in vitro cell binding in different cell line with different target expression. And together with that, we were able to also estimate this chi value to be 100 and further utilize the binding prediction to correlate with functional assay readout. For example, when you look at the inhibition of phosphorylation, again, we have a few different cell lines that allow us to really correlate to the binding versus the function, and then looking into how the combination approach for the monovalent parental 2 molecule is actually not behaving as well. For example, shown here, right? This is the combination at the molar equivalent doses versus the bispecific, and then use this particular binding model together with the PK measurement to understand what caused it. So again, I'm not going to go into the detail, but I think this is one important mechanism-based study for you to really probe into the mechanism of action for an important molecule, which was still very novel back then, and to understand the importance that this is actually a very important design element that you can really optimize when you have a number of different candidates. By the way, this chi value, right, is considered to be ranging from like 100 to 10 to the 5th. When you look at IgG-based antibody, and you do have a lot of wide ranges, right, to really think about how can you incorporate this particular concept using flow and estimation by mathematical equations, right, to allow for better candidate selection. All right, so moving forward very quickly, one work I did at J&J is really this very interesting QSP-like model. The one thing I have to mention is that that's the first time I got to learn a little bit about a modified genetic algorithm, right? In this particular study, again, published by Peng et al. Prior to what I worked on this, is that they did not have any constraint of the parameter, which was over 25 parameter, and they only have slightly more data, number of data points, which makes the, I would say, the parameter estimation challenging. So what we did is that we want to have a little bit more confidence for how this model is able to predict, right, untested combination scenario. So what we did is that we recreate the model in NumM, And we searched about 10 parameters that we can find reliable measurement in terms of physiological value. We replace them and then allow the model to be further evaluated. As you can see, right, this is our model fitting versus the observed data across the different types of combination or single monotherapy treatment, and looking at different cell types, right, Treg, CTL, and also tumor growth. And I think this is a good example for really understanding what data you have, and also like when you create a model that has a lot of degree of freedom, I honestly don't feel that some of the parameter value, right, coming from this particular genetic algorithm was that trustworthy going through this exercise. So one of the final examples before I go to last example is that we further expanded our QSP approach, right, to understand a little bit more sophisticated, for example, non-small cell lung cancer related combination therapy, which were presented at ACOP back in 2017 as a poster. This study was also done in collaboration with Rosa. I think, at least from my perspective, right, this is a very good implementation in my labs in biology, really understanding some of the more complicated scenario, which again, right, if you look at a QSP modeling paper these days, you have much better model now compared to what we had back then. So my final case, which I promised some of my colleagues here, is this there's a very interesting longitudinal exposure response analysis together with the mechanism for body weight losing drug. So one thing that I was working on is really for to work on this long-acting GD15 molecule, which was going through the first human study. Actually, we were getting ready for that, right? And then we want to understand whether obese cynomolgus monkey is a translatable model using dulaglutide, which is the long-acting GLP-1 agonist, right? And then also this molecule that has not gone to human yet. We want to understand whether there's probability for success. So as you can see, we tested different drug level, and we also measured the food intake reduction over time, and also the body weight change over time. We also measure PK for both dula as well as this GD15 molecule. So the question really is, can we explain this, right? So as a lot of you guys know, Kevin Hall published the macronutrient model, which was very sophisticated. And fortunately, by working with Dave Polidori on this particular project, we were able to simplify the core component of the macronutrient model to the relationship between food intake, energy expenditure, the feedback loop, together with the PK/PD built into the model, right? For example, looking at the Emax model or the dulaglutide model, almost good. And also the human prediction, what we did is that the model was able to capture the population level changes, right? In terms of the food weight reduction as well as rebound, body weight changes, and also the PK, right? And also this is dulaglutide. And we were able to predict what happens, right? Using literature data for liraglutide and also semaglutide, and understand the body weight change potential for this new molecule, new mechanism of action. I think one thing which I don't have time to talk about here today is that this model, right, allows us to really show that this molecule really works on body weight loss, right, without increase in energy expenditure, because there are also molecules, for example, like ostomodulin, right, that also changes potentially energy expenditure. And the most important thing, this model has the potential to really be incorporated, right, into a more of a human study, and also allow for prediction for long-term body weight change using the initial 12-week time, which I did, right, which was not published. But again, I think it's a very capable model which allows for model fitting. All right, I'm gonna quickly end here just to talk a little bit about the learnings, right? I think there are a lot of learnings along the way. I think the most important thing, right, is I want to talk about the importance of great mentors, and without really working with them, without them helping you or helping me, right, I don't think any of the work could be possible. And I would really say that work very closely with your peers, because ultimately, right, they are your best ally. And I think troubleshooting with them, with your mentor, is probably one good learning experience. And ultimately, right, really train and share your knowledge with people. Be open-minded. I actually wrote this down. Be open-minded, optimistic, curious, adaptable, and enjoy the process, right? When I was preparing the deck a few days ago, and that was touched upon actually even like day one and yesterday. I think, again, right, I'm going to just quickly touch upon this reverse translation, forward translation cycle, which I thought about, right, for a little bit. Really, this is the way to do it. And then I want to give a lot of acknowledgement, right, to Baitem, my internship advisor, my PhD advisor, Kentamo Danny Shen, actually, I'm a committee, right, Nina Josh, and also Tony Blau and Chris Miller, who was my undergraduate researcher at UW Medicine, and also, of course, at UF for my postdoc, Stefan, Larry, Puneet and Shilin, who were the first and the third postdoc with Stefan, and also Ping Zhao, which I didn't talk about the work I did with him at the FDA for the PBPK work. And of course, a lot of people at Janssen, particularly Wei Rong, who really trained me in terms of thinking through a lot of problems. And of course, right, the key contributor and also my current colleagues at Adagene, who I didn't get to talk a lot about the examples there, but I think at least I want to give the overall journey through several different types of model. Okay, I think I'm done. Pretty good timing. Thank you very much.
Thank you, Ruben. Any questions?
Thank you so much for your presentation. It was a lot of information in a very short period. But something that it catch my eye is more like a comment. So there is like a more pragmatic solution when you're working with soluble target, and the turnover rate is very fast comparing with the half-life of the antibody. When this is happening, when the soluble target is bound to the antibody, it's going to change the clearance, it's going to follow the clearance of the antibody. So because of that, you are going to see, if you are quantifying total soluble target, you are going to see a accumulation of the soluble target. And then based on the accumulation of the soluble target, you can extrapolate or you can estimate the soluble target engagement.
Yes.
And actually, it works very well, and it's a very, very simple model. So sometimes we don't need to do these mechanistic models or TMDD models. So.
Yeah, thank you for the comment, right? So you're absolutely right. The synthesis rate of the soluble target can be very well estimated if you push the dose high enough to really understand the initial slope, that rise. I think really, again, right, we have gone through enough examples, right, at J&J to really show that's a good way to really constrain the parameter. I think the IL-6 example, right, again, we have just very robust measurement for the 3 different analytes. That gave us a little bit pause, because I can tell you, right, the initial rise of the total target concentration is great. But I think it's still having the measurement, right, give you additional resolution for understanding the parameter a little bit better. But I think for the pragmatic use, just understand generally where things are, your approach makes perfect sense.
So usually our objective is to quantify the target engagement. So because of that, I'm always trying to convince everybody that just quantifying total soluble target and also total PK, total antibody is sufficient, because if your target is soluble, like I said, based on the accumulation, also case by case, but if you have an accumulation of the soluble target, then you can calculate target engagement, so it's sufficient. And if you have nonlinear PK because the target is a cell surface receptor, then you don't even need to quantify receptor occupancy, because just based on nonlinear PK, you can extrapolate the receptor occupancy.
Yes,
so.
yes. Yeah, that's why the TNF-alpha example worked. We don't have measurement for the free TNF-alpha, but it was working fine. I agree with you completely. Thank you.
Ben, you promised us an example, so I need to ask that question. So great talk, a lot of examples. I'm really impressed with all the work that you have done. For the last example of GDF15, it's remarkable that, you know, how you used Kevin Hall model to implement the energy intake and the behavioral change. But the thing is that using the mice model, you predicted the human data, but what we have seen...
It's monkey, not mice.
Oh, sorry, monkeys, yeah. But what really happened in that field is that all the clinical data actually were kind of disappointing, right? So any thought, you know, did you do any post-mortem to understand that why it happened? You know, what are the things that you could have changed, or moving forward, what will you change in this translation?
So that's a very good point, right? So if you go into this particular paper, we actually showed what monkey and human are similar in terms of those parameters, because we were able to pull, right, the parameter value coming from Kevin Hall's paper. They actually look into this, right, using human data for like some of the older, like even like a caloric restriction study, right? Because those are more physiological. And I can tell you that this particular obese monkey model, even though we say it's obese monkey, they're still pretty lean compared to obese patients, right, human. There are physiological differences in terms of some of the parameters. Again, I didn't include here, but you're absolutely right. I think this model, even though it can predict dulaglutide fairly well using the related, right, food intake change and also the body weight loss, this is dulaglutide. I think the jury is still out there. Can it do a good job in terms of understanding, for example, like the more advanced, right? Like when you really hit the body weight loss more than 15%, and do we have enough data right now, right, to look into some of the more well-defined calorie restrictions, like a food intake study, right? Because when we look at the semaglutide and liraglutide study back in the days for the human study, they were very small, very like a confined environment. It's not real world. And I think that's where, but from my perspective, what I can tell you is that when this model was initially developed, right, I actually used the semaglutide data to actually do a little bit more than what I showed here. It was able to capture some initial data. I didn't do this for the most current data and also for like some of the GIP, GLP-1, right, type of molecule. But I do think that this model has potential. It needs to be further verified.
Okay, thank you, Dr. Ben, for the great presentation. Okay, now we